Hello and welcome back. This is the second video in our video series for chapter 2. And if you're ready, let's get right into it. So this subtopic is 2.2 Structure and Function, Cell Membrane and Organelles. So we will talk about the plasma membrane and also the other organelles that are present in the cells. Before we go into the lesson, I would just briefly talk about the learning outcomes. I hope that by the end of this lesson, you will be able to show the detailed structures of typical plant and animal cells. I hope that you'll be able to explain the structures and functions of the following organelles, which are nucleus, and that you should be able to show the structure of plasma membrane based on the fluid mosaic model. Last but not least, you should also be able to explain the structure of the plasma membrane and the function of each of its components. Okay, so let us look at the cells that we are going to dissect. Today's lesson, or this 2.2 lesson, we are going to focus on eukaryotic cells, which are animal cells and plant cells. I would like you to draw animal cells and plant cells at the end of this lesson, but please do not draw the 3D version. I mentioned this in the Google Meet and I'll mention it again. When I ask you to draw, I don't want you to draw this or this. They are both accurate diagrams, but it's not necessary for you to draw such an artistic interpretation. Cukuplah kita gunakan diagram yang simple, yang 2D, yang saya akan tunjukkan di hujung video. Okay, so we are going to look at the organelles, or what you can say organelle is organ kecil. Okay, the smallest organs that exist in your cell. If you say your stomach, perut saya adalah organ. Organelle is the very small things that exist inside my cell. So example is mitochondria. Okay, itu adalah organ kecil. So kita akan tengok dan kita akan bandingkan animal cell dengan plant cell. So we will start with the most basic organelle, which is the nucleus. This is the largest organelle in your cell. It consists of a nuclear envelope. Envelope macam sampul surat. Okay, so it's something that cover the outside. Nuclear pores, which control the exit of substances such as RNA and ribosome from the nucleus. So ini adalah liang-liang ataupun lubang-lubang pada nuclear envelope. You also have the nucleoplasm, which is the interior of the nucleus. Apa yang nampak macam purple jelly-jelly di dalam, itu adalah nucleoplasm. And the nucleolus, the dark region of the chromatin involved in making chromosome. So bahagian yang paling gelap, involved in making ribosome. Bahagian yang paling gelap ini adalah terdiri daripada chromatin yang tebal dan kelihatan lebih gelap. Chromatin is we call the DNA when it is being stretched out. Okay, kalau DNA kita tidak kasih gumpal dia, kita kasih biar dia terburai, itu adalah chromatin. So the function of a nucleus is it contains DNA carrying genetic information that can be passed from one generation to another. How to draw a nucleus? You don't have to draw like this. You can draw with my guidance in my style. So we can start drawing a nucleus by drawing a big black dot. And then we draw one of this and we draw the nuclear envelope. When you draw the nuclear envelope, you leave a little space like this, and like this, and like this, and like this. If you want, you can color in the nucleoplasm. So this is your nucleus. When you draw anything, don't forget to label. So here I label the nucleolus, I label the nucleoplasm, I label the nuclear envelope, and I label the nuclear pore. That's it. That's my nucleus. And I am moving on to the next organelle. So the next organelle that we'll talk about is ribosomes. Ribosomes are the smallest and most numerous organelle. They are composed of protein and rRNA. dia terdiri daripada 70% rRNA dan selebihnya terdiri daripada protein. So these are manufactured in the nucleolus of the nucleus. Okay, ribosomes are created in the nucleolus. Some are found freely and some are attached to the surface of endoplasmic reticulum. Ada yang terapung-apung di dalam cytoplasm, ada yang melekat kepada organel lain yang digelar sebagai endoplasmic reticulum. Dan biasanya kita akan jumpa ribosom dia terkumpul dalam kumpulan. They are often found in groups called polysomes or polyribosome. Poly meaning many. The function of ribosomes are very simple. They synthesize proteins or polypeptide in the translation process. Okay, so if you want to draw the ribosome, you can draw a dot. Okay, boleh lukis saja titik macam ni. Boleh lukis banyak titik macam ini. Ataupun kamu boleh lukis dia punya structure di mana kamu boleh tunjukkan large protein subunit, large rRNA. Sorry. You can draw the structure where you show the large subunit and the small subunit, like this and this. And don't forget to label. And then, of course, I want to label and write ribosomes. Okay, moving on. The next organelle that we are talking about is the rough endoplasmic reticulum. Ataupun kamu boleh pendekkan dia sebagai RER. What is the rough endoplasmic reticulum? It is a membranous system of interconnected flattened sacs. Sacs, S-A-C-S, kalau kamu belum tahu, dia bermaksud kantong. Okay, so these are like bags or sacs that are connected and flattened. Dihimpit dan diratakan dan yang masih berhubung. The rough endoplasmic reticulum is usually continuous with the nuclear envelope. Means dia biasanya bersambungan dengan nuclear envelope. These flattened sacs are also called cisternae, and they are studded with numerous ribosomes which give it a rough appearance. Di permukaan rough endoplasmic reticulum ada ribosome. Tadi saya cakap ribosom kadang-kadang terapung, kadang-kadang dia melekat dengan endoplasmic reticulum. Inilah dia. Bukannya kita sudah pernah sentuh dan rasa dia kasar, tetapi bila kita tengok melalui mikroskop, dia nampak seolah-olah permukaan dia kasar disebabkan adanya ribosom. That's why we call it rough endoplasmic reticulum. Okay, so that's why we call it rough endoplasmic reticulum. And the function of RER is the site for protein synthesis. Usually, it will transport the protein that are made by ribosome. So, ribosome tolong dia buat kerja, ribosome tolong hasilkan protein, rough endoplasmic reticulum tolong hantar. If you want to draw, you can draw like this. Okay, pastikan kamu ada lebih dari satu layer of sacs. Pastikan dia bersambung. Dan kamu ingat untuk letak banyak-banyak ribosome. Remember to draw the ribosomes. Okay, I'm going to do the animation again. And then you label it as rough endoplasmic reticulum. Sekian. Next is the smooth endoplasmic reticulum. The smooth endoplasmic reticulum is a membranous system of interconnected tubules. They have an outer surface that lacks ribosome. Permukaan luar dia tiada sebarang ribosome. Itulah dia nampak seolah-olah macam dia ini licin. The function of the smooth endoplasmic reticulum is different from the rough endoplasmic reticulum. Tadi rough itu berkaitan dengan protein-protein saja. Untuk smooth endoplasmic reticulum, it is the site for lipid and drug metabolism. That means if you have any lipids or any drugs in your cells, this guy, this smooth endoplasmic reticulum, is going to help you to break down the lipids, break down the drugs. At the same time, it also synthesizes the lipids, phospholipids, and steroids. Dia bukan saja menghancurkan, dia juga menghasilkan lipid, phospholipid, steroids. And they transport materials. Dia boleh juga menghantar barang-barang. The materials are usually lipids and carbohydrates that are needed by the cell. Okay, let us try to draw the smooth endoplasmic reticulum. Are you ready? Okay, siap. Smooth endoplasmic reticulum. Moving on. Next organelle is the Golgi apparatus. So this one organelle has several names. You can call it Golgi apparatus, Golgi complex, Golgi body. Ketiga-tiga nama pun betul. Tapi pastikan Golgi G dieja dengan huruf besar. Sebab G adalah kata nama khas saintis yang jumpa bernama Golgi. This is one of the membrane-bound organelle, and they are made up of a series of flattened, stacked pouches called cisternae. They are also located in the cytoplasm, and usually you can find them next to the endoplasmic reticulum and near the cell nucleus. While many types of cells contain only one or several Golgi apparatus, plant cells can contain hundreds of these kinds of Golgi apparatus. Okay, so what is the function of this strange thing? The function of the Golgi apparatus is a little bit like a factory. Okay, kamu tengok dia punya fungsi lebih kurang macam kilang. Kita boleh umpamakan dia dengan kilang. They are responsible for modifying, packaging, sorting proteins and lipids into vesicles and then transporting the vesicles to target destinations. So kita tengok dia punya fungsi adalah untuk lepas dia terima, dia akan mengubah suai, modify, dia membungkus, package, sort, susun ataupun susun atur, sort them into vesicles and then transport the vesicle to target destination. Kemudian transport menghantar. So dia terima, dia mengubah suai, dia bungkus, dia susun dan dia hantar. So a lot like a factory. And if you want to know, it has a cis phase and a trans phase. What does it mean by cis phase and trans phase? Bahan-bahan akan masuk melalui cis phase yang menghadap endoplasmic reticulum dan barang-barang yang sudah modify sedia untuk transport akan keluar melalui trans phase. Trans because it is already transformed. And this trans phase is usually positioned near the plasma membrane of the cell so that it can be pushed. Okay, the modified and the processed products such as proteins will exit in the form of smaller detached vesicles. So, mungkin kamu ada nampak sudah perkataan vesicles beberapa kali. Kalau ada yang belum jelas, vesicle ini kamu boleh bayangkan dia macam bubble. It's like a bubble that can be empty or it can contain substances and they move around in the cytoplasm. Okay, just imagine they are bubbles. Okay, so let us start by drawing this Golgi body, starting with the cis phase. We start with layer number 1. Okay, cisternae number 2, number 3, number 4, number 5, number 6, and then I draw an arrow to show that materials go in from the cis phase and they come out from the trans phase. Okay, here I write trans phase. I draw some vesicles to make it clear that there are materials coming in and coming out of this Golgi body. And then I label Golgi apparatus. Okay. Let us move on to the next organelle. The next organelle is the lysosome. So this is a very small membrane-bound vesicle containing hydrolytic enzymes. These are secretory vesicles produced by the Golgi body. So biasanya lysosome ini adalah organelle yang mengandungi enzim-enzim untuk menghadam sesuatu. Dia boleh dihasilkan oleh Golgi body. So what is the function of lysosome? Function of lysosome is to carry out intracellular digestion in a variety of circumstances by phagocytosis. Contoh, kalau kita punya sel makan sesuatu, okay, makanan sudah berada dalam sel, yang akan menghadam makanan itu adalah enzim-enzim dalam lysosome. You can also use the lysosome to undergo autophagy. Autophagy means that your cell is eating part of itself, or it recycles part of its own organic material. Contoh, dalam sel badan saya ada mitokondria yang tidak berfungsi sudah. Okay, it's old mitochondria, it's not functioning properly. So my cell will take that mitochondria, use the enzyme from the lysosome, and digest the old mitochondria. Self-digestion can also occur if you have excessive leakage from a large number of lysosomes. Okay, you can also use the lysosome for self-digestion. Or it can happen accidentally. So if you want to draw, just draw a circle and then draw this circle inside. The smaller circles inside the bigger circle is the hydrolytic enzyme, or you don't have to label it and you just write lysosome. And then we can move on to our next organelle. Alright, the next organelle is something that we are all very familiar with. It is the mitochondria. So, ini adalah organelle paling biasa kita cakap. Ini adalah mitochondria. Mitochondria is surrounded by a double membrane. You have an outer membrane which is simple and quite permeable, means bahan-bahan senang untuk keluar masuk. And you have the inner membrane, which is highly folded into cristae, which gives a large surface area. The inner membrane of the mitochondria has a fluid region inside called matrix. There are lots of enzymes that can be found on the surface of the inner membrane, and the mitochondria has its own DNA and simple ribosomes. Why? Later you can find out why. But basically, you should know that the mitochondria does have its own DNA, and it does have its own form of small, simple ribosome. Okay, the function of the mitochondria, if you don't already know, is it is the site of aerobic respiration, or you can also say it is the site of ATP synthesis. Energy is produced in the mitochondria. If you're ready, let us draw this mitochondria together. So, I'm starting with this shape. This is the outer membrane, and then I'm drawing the inner membrane. Okay? Pastikan inner membrane kamu ada lipatan. So, dia ada lipatan. Banyak kah, sedikit kah, asalkan ada tunjukkan lipatan. And then I label the outer membrane. I will label the inner membrane. And now I want to label the matrix. Okay? Ruangan di dalam ini kita panggil sebagai matrix. And these folds, setiap lipatan yang kamu lukis itu, setiap lipatan dipanggil sebagai cristae. And this region between the outer membrane and the inner membrane is called the intermembrane space. And this is how you draw mitochondria. I hope you don't have any problem with this drawing, and I shall move on to the next organelle. The next organelle that we talk about is the chloroplast, what makes plants green. So the function of the chloroplast is as the site for photosynthesis. How is the structure? Chloroplast also has double membrane. Chloroplast also contains chlorophyll and other photosynthetic pigments. Inside the chloroplast, when you look inside, you will see the thylakoid. These are membranous system in the form of flattened interconnected sacs inside the chloroplast. If you put many thylakoid together, you can call that a granum. You have more than one granum, you call it grana. And the region over here is called the stroma. It is a fluid region that contains chloroplast DNA, ribosomes, and enzymes. That's right. Chloroplasts also have their own DNA, their own ribosome, and their own enzymes. Is this a conspiracy theory or a very well-known biological fact? That's up for you to learn, Google, and discover. Okay, so the stroma contains chloroplast DNA, ribosomes, and also enzymes. Okay, let's draw a chloroplast. Okay, if you want to draw chloroplast, remember it has two membranes. Okay, dia double membrane organelle. So, lukis dua macam ini. And then you draw one thylakoid. Okay, I label this as a thylakoid. And then I draw more thylakoid. I draw many. Satu stack macam ni, saya panggil sebagai granum. Bila banyak, saya panggil sebagai yes. And then this region is called the stroma. This is where you find the other stuff. And this is called your chloroplast. Remember, photosynthesis occurs on the thylakoid membrane. Okay? Bahagian thylakoid, which is the garis-garis, di sana lah photosynthesis berlaku. Oh, sorry. Okay. Alright, let us move on to the next organelle. The next organelle is the centriole. The centriole is a pair of short microtubules. They are arranged at right angles to each other, and each of these centrioles are made up of 9 sets of 3 microtubules. Apa yang dimaksudkan dengan 9 sets of 3 microtubules? Kamu tengok dia punya susunan ini. Kalau kamu boleh zoom in gambar kamu lah, okay? You look up here. Okay. You look up here. Dia adalah 1, 2, 3, susun 1 kali, 1, 2, 3, susun 1 kali, and that repeats for 9 times. And you can also see that sometimes the centriole will have spindle fibers. Gentian gelendung, okay, spindle fiber come out from the centriole. When it has spindle fiber coming out, you can call it as ester, you can call it as a centrosome. Or centrosome is a region that contains centrioles. Okay, apa function centriole? Saya ada cakap spindle fiber, saya ada cakap BM dia gentian gelendung. Centrioles are involved in cell division in animal cells. Dia tiada dalam plant cell. It will initiate the spindles that organize and separates the chromosomes. Bila kita masuk dalam chapter 3, which is mitosis meiosis, we will talk more about the centrioles. Now let us learn to draw the centrioles. Okay, centriole. You can draw it in this way. Tidak semestinya perlu lukis cara dalam slide sebelum ini, sebab cara slide sebelum ini dia sangat complicated, merumitkan keadaan. Saya okey saja kalau kamu lukis kamu punya centriole macam ini. Kalau kamu mau, kamu boleh tambah itu spindle fiber, itu gentian gelendung, but if not, no need. Okay? Kalau tidak lukis pun tidak apa. Kalau mau, kalau rajin lukislah macam tu, tapi tidak penting sangat. Okay? So don't waste your time. Okay, so that is it for centrioles. Let us move on to the next organelle, which is the vacuole. A vacuole is membrane-bound sac that contains water or dilute solutions of salts and other solutes. And the central vacuole in plant cells are filled with cell sap. Dalam arti kata lain, dia cumalah satu kantung yang mengandungi air ataupun bahan-bahan yang terlarut yang lain. Why? Why do we have vacuole? Vacuoles are very important in keeping the plant cell rigid or turgid. Dia memastikan sel tumbuhan itu dia tidak lembek, okay? That it will remain turgid, tegang. And some unicellular protists like amoeba will have food vacuoles for digesting food and contractile vacuoles for expelling water. Okay, so vacuole ini boleh, bukan saja air lah, dia boleh mengandungi bahan-bahan lain juga. Tengok sel yang kamu sedang kaji itu. How to draw vacuole? First you draw cell, and then you draw vacuole, and then you label vacuole. Begitu saja. Now, if you are ready, let us try drawing the animal and the plant cell together. Okay, we are going to start with this animal cell. I draw the plasma membrane, and then I draw the nucleus. I draw the nuclear envelope, and then I draw the rough endoplasmic reticulum, and another rough endoplasmic reticulum, another rough endoplasmic reticulum, and a smooth endoplasmic reticulum. I draw the Golgi body over here with the vesicles, and I draw some mitochondria, some lysosome, some centriole, and I add the rest of the ribosomes. And then after you draw something, it's a good idea. Oh, sorry, I forgot to draw one more flagellum. Okay. After you draw something, it's a good idea to label. So I am going to label the nucleus, label the rough endoplasmic reticulum, also called RER. I label the mitochondria, I label the Golgi apparatus, I label the vesicle, the ribosome, the centriole, the smooth endoplasmic reticulum, ataupun SER. I label the flagellum and the lysosome and the plasma membrane. So these are all the organelles that you can draw and label in your animal cell. Nanti saya akan minta kamu lukis animal cell dengan plant cell. So pastikan kamu lukis 2D animal cell like this one. Susunan organelle dia berlainan tidak apa, yang penting kamu label apa yang perlu label dan lukis apa yang perlu lukis. We're not done here. We have one more cell to look at. So let us look at the plant cell. So we start with the plasma membrane. We draw the nucleus, the nuclear membrane, the cell wall, the rough endoplasmic reticulum, rough endoplasmic reticulum, rough endoplasmic reticulum, smooth endoplasmic reticulum, vacuole, Golgi body, mitochondria, another mitochondria, chloroplast, and another chloroplast. And of course, I would like to add lots of ribosomes. And then I'm going to label everything: nucleus, plasma membrane, cell wall, rough endoplasmic reticulum or RER, smooth endoplasmic reticulum or SER, mitochondrion, vacuole, Golgi apparatus, ribosome, chloroplast, and label it as a plant cell. Okay. So hopefully after that you are able to achieve learning outcome 1 and 2 of this subtopic. Now, if you realize, we talk about a lot of organelles, but we are forgetting one more important organelle, which is the plasma membrane. And we will talk in detail about the plasma membrane now. Okay, so what is the plasma membrane? I'm sure you already know. This is the layer on the cell, okay? Make sure that the cytoplasm stay in one place. The plasma membrane is a selectively permeable phospholipid bilayer that is found in all cells, whether they are prokaryotic or eukaryotic. Selectively permeable means it only allows certain molecules or ions to pass through it by simple diffusion and facilitated diffusion. Bukan calang-calang molekul boleh keluar dan masuk. Sesetengah sahaja boleh keluar dan masuk. Ikut apa yang plasma membrane benarkan. So when we talk about the structure of the plasma membrane, we usually go by this theory suggested by Singer and Nicholson. Singer and Nicholson adalah nama saintis yang mengkaji struktur plasma membrane. Dan model yang diorang sarankan adalah fluid mosaic model. When they say the word fluid, bila diorang cakap fluid in fluid mosaic model, they mean that the phospholipid bilayer and the proteins can move laterally or change places and thus give the membrane fluidity. Phospholipid, yang biji-biji yang ada 2 ekor ni, dengan protein-protein yang ada pada plasma membrane, dia boleh bergerak-gerak. It can move laterally, kiri, kanan, depan, belakang. And they can change places. Dia boleh bertukar tempat. So, ini memberikan plasma membrane kita fluidity. Dia seolah-olah dia bergerak. Bukan seolah-olah. Memang dia bergerak-gerak macam permukaan laut. And you can also have lateral movement of a phospholipid. You can also have the flip-flopping of a phospholipid. So, yang kiri, kanan, depan, belakang itu memang biasa. Yang atas terbalik pergi bawah pun boleh berlaku tapi pada kadar yang lebih perlahan. When we say the word mosaic, it refers to the various proteins that are embedded in the phospholipid bilayer, giving it a mosaic appearance. So, pada plasma membrane kita ada banyak protein yang terbenam. So, bila kita tengok protein-protein yang terbenam ini, dia nampak macam tiles lantai mozaik yang berwarna-warni, yang bermacam-macam jenis. Okay, so when they look at this, they say it looks like the mosaic tiles, which is why they say it is mosaic pattern. Kalau soalan tanya, apakah yang dimaksudkan dengan fluid dan mosaic? If the question asks what is referred to by fluid and mosaic in the fluid mosaic model, you use this and this explanation. Gunakan penjelasan ini dan ini. Okay, so let us move on and look at the plasma membrane components. What is in or on the plasma membrane? We have 4 main components, which are the phospholipids, proteins, carbohydrate chain, and cholesterol. What is phospholipid? Phospholipids, these are this thing. They are arranged in a bilayer. Dia disusun dalam 2 lapisan. Your phospholipid will have a polar hydrophilic phosphate head which face outwards. So, yang merah biji-biji ini adalah kawasan yang suka air. Hydrophilic. And you have the non-polar hydrophobic fatty acid tails facing each other. So, yang kuning macam kaki ini adalah non-polar hydrophobic fatty acid tails. Okay, so they will have this hydrophobic region in the middle. Why is it important that we have these phospholipids? Well, the phospholipid bilayer prevent polar solutes from diffusing across the membrane, but it allows the diffusion of hydrophobic molecules such as lipid hormones. That means polar solute cannot pass through right away. Benda yang polar tidak boleh terus limpas melalui phospholipid bilayer. Dia perlu melalui tempat lain. Tapi benda yang boleh larut dalam lipid, contohnya steroid, dia boleh terus limpas. Dia boleh terus melalui phospholipid bilayer. These phospholipids affords the cell the ability to regulate the movement of these substances via transmembrane protein complexes such as channel and carrier protein. What this means is, Kalau kita ada phospholipid bilayer, sel kita boleh kawal apa bahan yang masuk dan apa bahan yang keluar. Dia ibarat dinding. Dan untuk bahan yang besar, keluar atau masuk daripada sel, dia harus melalui protein complexes yang ibarat gate. Okay, so let us move on. We'll talk about the next component of the plasma membrane, which are the proteins. So we have two types of proteins in the plasma membrane. The first type are the peripheral proteins, which are not in the lipid layer. They are loosely bound to the surface of the protein. And we also have the integral protein, which penetrate the hydrophobic core of the lipid bilayer. And they are often completely spanning the membrane. Completely spanning membrane means that dia ada di kawasan ini dan dia tembus ke kawasan ini. Kedua-dua layer pun dia ada. Okay. The proteins that are in the plasma membrane have several functions. They can be there as transport protein. They can be there for enzymatic activity. They can function for signal transduction. They can be there for intercellular joining. They can be there as cell-cell recognition. And they can be attachment to the cytoskeleton and extracellular matrix or ECM. Okay, if you are not clear with what any of these mean, either Google it, Google Translate it, or ask me personally. What it means by cytoskeleton and extracellular matrix. Cytoskeleton ini adalah rangka untuk sel kamu. Like small skeletons, seolah-olah sel kamu pun ada tulang lembut. Okay, yang membantu sel kamu mengekalkan bentuknya. Okay, so a lot of cells have their own cytoskeletons that maintain or support the shape of the cell. Extracellular matrix means that you have something outside of the cell that your cell can hold on to. Nanti kita belajar pasal tulang dengan cartilage, kita akan tengok extracellular matrix. Okay. That was the second component of the plasma membrane. Now we are looking at the third component of the plasma membrane, which are the carbohydrates. The carbohydrates that we find on the plasma membrane are usually branched oligosaccharides ataupun branched carbohydrate chain. If the carbohydrate chain is bonded to a protein, we call it a glycoprotein. If it is bonded to a lipid molecule, we call it glycolipid. Tengoklah dia bersambung dengan siapa, itulah kamu kasih nama dia. Why is this carbohydrate on the plasma membrane important? These oligosaccharides, they vary from species to species, individual to individual, and even from cell type to cell type within the same individual. So, kita boleh gunakan carbohydrate chain pada permukaan plasma membrane untuk cell-cell recognition. Recognition, recognize, mengenal pasti, perkenalan. Sel-sel kita boleh menggunakan carbohydrate chain pada permukaan plasma membrane untuk kenal sama ada sel yang saya jumpa ini sebahagian dari badan yang sama atau tidak. Ataupun sel ini adalah kuman yang berbahaya, kuman yang okey, ataupun benda lain. Cell-cell recognition is the ability of a cell to distinguish one type of neighboring cell from another. Okay, is this one of us or is this enemy? This is important in cell sorting and organization as the tissues and organs in development. This cell-cell recognition is also the basis for rejection of foreign cells by the immune system. Like I said, this is how your body knows whether it is looking at a friend or an enemy that it needs to kill. Cells will recognize other cells by keying on surface molecules, often carbohydrates, on the plasma membrane. Alright, and last but not least, we'll talk about the fourth component of the plasma membrane, which is cholesterol. Okay, cholesterol, the lemak that usually contributes to heart attacks, cardiovascular disease. That is the main risk when you are obese. Okay. Kolesterol bukan selama-lamanya jahat. Kolesterol ada kebaikan dia juga. Mari kita tengok antara kegunaan dan kebaikan kolesterol. So this cholesterol will be wedged between the phospholipid molecules in the plasma membrane. Dia akan disisip di antara phospholipid molecule dalam plasma membrane kamu. Why? The cholesterol is there to regulate membrane fluidity. At warm temperature, cholesterol will restrain the movement of phospholipid and reduces fluidity. When the temperature is warm, your plasma membrane tends to move more. So the cholesterol will tell your phospholipid, don't move so much. Chill, bro. And when you reach cool temperature, cholesterol will maintain fluidity by preventing tight packing. When it is cool temperature, your phospholipid want to come together very close and not move at all. When it is cool, cholesterol will make sure your phospholipid are not too packed. Dia akan pastikan dia ada penjarakan sedikit supaya kamu punya plasma membrane masih boleh bergerak, masih fluid. Okay, so what I've said, all that about the four components of the plasma membrane, the fluid mosaic model, is basically what you see in this diagram. Now I want you to draw this diagram. I don't want you to draw it 3D like this. I want you to draw 2D because the 2D version is easier. So let's draw. We're going to start by drawing some proteins. Okay, I am drawing one protein here, another protein here, another protein here. I also draw one more protein like this. And then I will draw the phospholipid bilayer. So I start with the phospholipids. Okay, sini saya lukis beberapa biji. Saya lukis lagi satu layer. Lukis lagi satu layer. Sini saya mau tambah satu cholesterol, so I draw one cholesterol here. And then I draw the rest of the lipid. I want to add another cholesterol, so I draw another cholesterol here. I add the lipids, and lipid. Okay. Something else I want to add is the carbohydrate chain or the oligosaccharides. So I draw one carbohydrate chain attached to a protein and one carbohydrate chain attached to a lipid. Now I'm going to label everything. This side with the carbohydrate chain is the extracellular matrix ataupun bahagian luar sel. This area inside is the cytoplasm, bahagian yang tiada carbohydrate chain. This is my phospholipid bilayer. This inside yang ada garis-garis kaki-kaki ini adalah hydrophobic region. Di luar adalah hydrophilic region. I'm going to label the integral protein. Asalkan protein itu dimasukkan dalam phospholipid bilayer, dia terbenam sedikit pun dalam phospholipid bilayer, kita boleh gelar dia sebagai integral protein. And this one that is sticking out is called peripheral protein. This is cholesterol. Pastikan ejaan kolesterol kamu betul. Dia bukan kolesterol. Dia kolesterol. Okay, so make sure you don't forget the E. Kalau kamu mengeja integral protein, dia adalah INTEGRAL. Jangan kasih extra R. Right, and then we label the glycoprotein. We label the glycolipid, and you can also label it like this. Kalau ada label yang macam ini, means dia tengok bahagian carbohydrate chain sahaja, ataupun kita boleh gelar sebagai oligosaccharides. Okay. So that is all for 2.2. Quick summary. In this 2.2, we talk about animal cells, we talked about plant cells, and we talk about the organelles. We talk about the structure. We talk about the function of each organelle. We talk about the plasma membrane and the fluid mosaic model. Hold on, let me do that animation once again. Talk about the fluid mosaic model. Okay, so what should you do now? Now, you should take out a piece of paper and draw a prokaryotic cell, a eukaryotic animal cell, and a eukaryotic plant cell. Label the cell and its organelles. Next, in a table, you should write the similarities and the differences between the prokaryotic cell and the eukaryotic cells. And number 3, draw and label the plasma membrane according to the fluid mosaic models. Please submit your drawings and tables in the Google Classroom. I think I've already provided you with a place to submit. So yeah, that is pretty much it. Thank you very much for sticking with my lesson. This was quite a long video, and I hope to see you again soon. Ciao for now.